So now I want to introduce the first speaker of the day. He's Vivek Keshri. He's an assistant professor at REVA University in Bengaluru, and he has been interested in characterizing the diversity and functional evolution of beta-lactamase protein families, and he will share the research that has been conducted in the previous years in this topic. So I will give you the word, Vivek. Yeah, and this will be a 30 minutes talk, and you are encouraged to make all your questions in the Q&A box. That is the one that we are going to monitor to ask the speaker later. But you can also chat with other participants of the events in the chat, and maybe sharing from where you are joining us today. But just keep in mind that the questions should be in the Q&A box. So thank you, Vivek. It's yours, the word.
Thank you, Henry. Good morning, everyone. My name is Vivek Keshri. It's my great pleasure to talk about my research work at this International Symposium for South Asia. Presently, I'm working as an assistant professor at REVA University, Bengaluru. Throughout my career, I have been passionate about the bioinformatics data analysis, and it is a really true honor to share my research work with this symposium and distinguished audience. Before we delve into the heart of my presentation, I would love to say thanks to the organizer for organizing this international symposium, where experts from the different background can come together to share their ideas, research work, and experiences. In the following slide, I will be talking about the general background of the beta-lactamase and antibiotics, beta-lactam antibiotics. And based on the review of the literature, I will share my objective of the project, what are the objectives I decided, and based on some tools and technique in bioinformatics approach, how I implemented the all methodology to achieve the all goals or objective, and followed by I will show some slide related to the results. And based on the result, I will conclude my project. So let us concentrate about the antibiotic and beta-lactamase enzyme. So we know that antibiotics like beta-lactam antibiotics, like penicillin, cephalosporin, carbapenem, monobactam, it is a very important antibiotic which is used by the global health sectors. And the antibiotic is targeting and killing to the bacteria, inhibiting the growth of the bacteria. And in our society, countless lives have been saved by the antibiotics in medical history. And because of the antibiotic, our medical field is very advancement, and all antibiotics are classified or grouped into two major categories: broad spectrum and narrow spectrum antibiotics. Actually, whenever antibiotic is targeting or killing wide range of the bacteria, that is known as broad spectrum antibiotic. But whenever antibiotic is targeting or effective on specific some of the bacteria or limited range of the bacteria, then that is known as narrow spectrum antibiotics. Apart from all facilities, all the benefits of the antibiotics, the major global health concern is antibiotic resistant spreading all over the world. The antibiotic resistant is spreading through different mechanisms like overexpression of the efflux pump, low porin permeable structure in the bacterial cell, and healthcare setting, like in hospitals, a lot of chances to spread the antibiotic resistance genes or bacteria from one person to another. And in our subpopulation or population, like in our community transmissions, the antibiotic resistance is spreading because it is a communicable disease. And not only, it is not only limited in one nation or in one subpopulation, but because in our community, a lot of people, they are traveling from one nation to another. So like global travels is also important. One causes of for the antibiotic resistant spreading in all over the world. And in this presentation, I'm going to talk about the beta-lactamase enzyme mechanism, how it is responsible for the antibiotic resistant and spreading, and their diversity, followed by how beta-lactamase function evolved through different mechanisms. So we know about the antibiotic like penicillin, cephalosporin, carbapenem, monobactam. All these antibiotics are well known. And the common structural feature of these antibiotics are beta-lactam ring. You can see that in every antibiotics have a common structure, which is known as beta-lactam ring. And bacteria, different bacteria, they are producing beta-lactamase enzyme. So beta-lactamase enzyme is hydrolyzing or breaking the structure of these beta-lactam antibiotics, which is breaking the structure of beta-lactam ring in every antibiotics. So beta-lactamase, as whenever a beta-lactamase enzyme is hydrolyzing the structure of antibiotic, then antibiotic become ineffective. It does not work on the bacteria. That's why beta-lactamase enzyme is responsible for the resistant. The major question is if beta-lactamase is responsible for the resistance, so how many beta-lactamase enzymes are well known in our scientific community? So if you check some recent articles like last 20 years or 10 years or 5 years, so many researchers, they reported that how more than thousands beta-lactamase enzymes are well known in our scientific community. And all these beta-lactamase enzyme is classified into two major categories: serine beta-lactamase and metallo-beta-lactamase enzymes. So serine beta-lactamase is further classified into different classes like class A, C, D. And this classification is completely based on the sequence similarity or sequence similarity or homology of the sequences. And the metallo-beta-lactamase, this is known as metallo-beta-lactamase enzyme because during the work of the beta-lactamase enzyme or during the mechanism of the resistance, this beta-lactamase enzyme is using the gene code, and that's why it is known as metallo-beta-lactamase. And these active residues in serine beta-lactamase enzyme is serine residues. That's why it is known as serine beta-lactamase. But instead of serine, in case of metallo-beta-lactamase, there is a histidine, and it is using zinc ion. So more than thousands beta-lactamase enzyme are well known in scientific community. But the next major question is these beta-lactamase, more than 1,000 beta-lactamase enzyme, where they are present, only in bacterial domain or different domain of the life. We know that our living life is divided into three major groups like bacteria, archaea, eukaryotes, and nowadays many researchers, they are saying that virus is also a fourth domain of the life. So if you check the 20 years ago, one article published by the Martin and Koenig, and they clearly mentioned that the beta-lactamase are absent from the archaea domain. Similarly, some article reported related to eukaryotes that beta-lactamase is absent from the eukaryotic domain. So here we can see that in scientific community, they reported that beta-lactamase enzyme is producing only by the bacterial domain, not the archaea or eukaryotes. As a result, I set our objective of the project is to explore the beta-lactamase diversity in different domain of the life in archaea, eukaryotes, including the metagenomics also. And followed by the functional evolution of the beta-lactamase. So with the help of some bioinformatics pipeline, we can try to identify the novel beta-lactamase like sequences from different domain of the life. So because we know that in bioinformatics field, generally all researchers, they are using the BLAST analysis for sequence similarity search. But in my project, I didn't use the BLAST analysis. Instead of simple BLAST analysis, I used ancestor sequence reconstruction and HMM profile search in different databases like NCBI NR database and human microbiome project database and metagenomic database. So the hypothesis of this ancestor sequence and HMM profile search is because these two methods are very sensitive compared to the BLAST analysis, and these two methods can detect the remote homologous sequences from databases. That's why I created the ancestor sequence of beta-lactamase enzyme and HMM profile also to search in different database. And this hypothesis I published in Genome Biology and Evolution. And in this paper, I clearly mentioned about one flow chart, and it is illustrating the complete analysis pipeline. And here, in this flow chart, I mentioned that Argonaut database. So I downloaded the all beta-lactamase sequences, amino acid sequences from antibiotic resistant gene annotation database, followed by I created the ancestor sequence of different class of the beta-lactamase A, C, D, and B separately. Similarly, I created the HMM profile search, and after that I searched these ancestor and HMM profile in different databases, followed by some threshold criteria. I filter out the all unwanted sequences, who are the similar sequences, and followed by I created the comprehensive phylogenetic tree of the all beta-lactamase family separately. So I created the comprehensive evolutionary analysis. I created the phylogenetic tree of the complete beta-lactamase enzyme with the help of extent as well as novel sequences also. So here in this tree, you can see that all phylogenetic tree I created separately, class A, C, D, and all these are related to the serine beta-lactamase. And here the most important thing is in scientific community, more around 1300 to 1400 beta-lactamase is well known in scientific community. But in our putative data analysis in bioinformatics, bioinformatic data analysis, this ancestor and HMM profiles, we found more than 44,000 beta-lactamase like sequences from different domains of the life. And I created the comprehensive phylogenetic tree of a different class. And here I indicated that this is A1 clade. So in A1 clade, all these sequences are novel sequences, and it is a new clade in the phylogenetic tree. So it is indicating that the ancestor sequence and HMM profile search is detecting the remote homologous sequences from different domain of the life. And these sequences belongs to human microbiome project, metagenomic, a lot of sequences from metagenomic, as well as archaea beta-lactamase sequences also. So in this study, we found that the class A, class C, and class D new beta-lactamase like sequences also present in bacteria, metagenome, and eukaryote. But in this paper, I mentioned only archaea and metagenome. And based on the analysis of metallo-beta-lactamase also is the same situation. We found numerous beta-lactamase sequences from metagenome and ARCHIA also. So based on this identification of beta-lactamase enzyme, we concluded that the beta-lactamase are more diverse than currently understood. And a lot of sequences, beta-lactamase like sequences are found in metagenomic database. So it is indicating that a lot of unannotated 2 beta-lactamase like is present in metagenomic, which is like we can say that metagenomic is a dark matter for the beta-lactamase enzyme. So this is the diversity of the beta-lactamase enzyme. Now I'm going to share one recent publication from Didier Raoult and Jean-Marc Rolland group, and major contribution of the Pierre Pontoroti. And this is the recent publication. And in this paper, the authors, they clearly mention that beta-lactamase, especially the metallo-beta-lactamase enzyme, is more diverse than previously understood in the scientific community. And now beta-lactamase enzyme is present in not only in the bacteria, but archaea, nanoarchaeota, humans, even in giant viruses also. So previously beta-lactamase is well known in bacterial domain of the life, but nowadays beta-lactamase is presently well known in a different domain of the life, like eukaryotes and archaea and metagenomics. And based on the bioinformatics data analysis, we cannot say that really beta-lactamase is present in a different domain of the life. That's why the recent group of the Didier Raoult, the recent publications, they picked the authors, they picked some sequences from different domain of the life, and they tested through wet lab experiment in the sequences, and they found that the beta-lactamase is really present in different domain of the life, like especially archaea and eukaryotes. So based on the wet lab experiment, they proved that beta-lactamase is present in different domain of the life. So it's more diverse. And if it is more diverse, then it is really a major global health concern about the beta-lactamase, that it is a major cause of the antibiotic resistant in our environment and the health sector. So this is the complete diversity of the beta-lactamase enzyme. Now I'm going to share some slide related to beta-lactamase function, how beta-lactamase function evolve. So in one paper published in Evolutionary application, and in this paper, I tried to map the genotypic information with the phenotypic data. Like genotypic is like, based on the amino acid sequences of the beta-lactamase, I created the phylogenetic tree. And here phenotypic information means I downloaded the all article of more than 1,000 beta-lactamase original article I downloaded, and I extracted the MIC related informations from the publication. And I mapped the all MIC information over on the phylogenetic tree based on the genotypic and phenotypic mapping through R programming. And in this mapping here in this slide, I can explain that here the blue color is indicating the functionally susceptible antibiotic, but in case of red color, it is indicating the resistant. And the green or aqua blue is indicating about the moderate resistant. It is resistant, but it is not highly resistant. So if you check this first figure, class A cefepime, so here we can see that the ancestor function, the extent function of the ancient function of the beta-lactamase is moderate resistant, but the extent, the present condition is completely different from the ancient function. And here we can see that some sequences are showing red color, means these sequences are these CTXM and beta-lactamase is resistant to the antibiotics. And these sequences are very closely related to these moderately resistant sequences. So it means that if two sequences are closely related, some here in this clade, if we consider this clade, some sequences are moderately resistant and some sequences are highly resistant. It means that their sequences are very similar because they belongs to the same clade, but according to their functional evolution, during the evolution, their function is changed. And if you check the distantly clad in this phylogenetic tree, then if you check this GES and TEM, these clades, so these clade is distantly located from the CTXM. So it means that if they are distantly located, then their sequence is dissimilar, but their function is same. It is also moderately resistant. But these function is different from the ancestor function. That's why we concluded that the function of the beta-lactamase is evolved through convergent manner. Similarly, in case of cephalothin antibiotic, is the same condition, like their extent function is different from the ancient, and some closely related sequences have a dissimilar function, even though they are sequences are similar, but their function is different. And some sequences have a similar function, but their sequences, amino acid residues, are different. So it is also indicating the functional convergent evolution. Similarly, in class B, piperacillin and other tree also. And based on this evolution study, we concluded that the functional evolution variation is not responsible, not originated by only single amino acid residues, because we checked the sequences of the all beta-lactamase enzyme, and we found that if one or two residues are dissimilar in the sequences, but their function is same, means if sequences amino acid residues only one or two residues are not responsible for the complete function of the beta-lactamase enzyme. And even though overall the study, we found that the convergently evolve these function. So these are the major study in this project. And because in our scientific community, a lot of databases are available related to the beta-lactamase enzyme, but none of them are presenting the complete comprehensive information about the sequence, structure, function of phylogenetic trees in a single platform or database. That's why we created a database based on the antibiotic resistant beta-lactamase enzyme. And in this database, we reported all sequences and the structures, function, and phylogenetic trees of the beta-lactamase of all classes, class A, B, C, D. And once again, thanks to the organizer and society, Indian Society of Evolutionary Biologists and the Society for the Study of Evolution. And some of my master students, they are searching for the internship. So if you have any vacancy or vacant position for the internship for my master, please let me know. And I'm interested in the collaboration also. And thank you very much for listening to me. And I will be happy to answer if you have any question. Yeah.
Thank you very much, Vivek, for the talk. It's impressive. My area of knowledge is a little bit different, but yeah, I enjoyed a lot learning about your research. I want to remind the attendees that you can drop your question in the Q&A box at any time. Even though if the talk is over, the speaker will be able to see the questions and reply privately. If you want to make a question as well, you can raise your hand and we will unmute you if you want to do it.
I checked the question and answer box, but there is no question in the box.
No.
So...
However, I'm seeing that there is a question on the chat, and we can read it for you. So,
sure.
As beta-lactamase enzymes in bacteria are continuously modifying themselves, are we thinking about any analogous molecules?
Yeah, like analogous molecule, I don't know exactly, but this is true that the sequences beta-lactamase enzyme is regularly modifying, and because of this major resistant bacteria are generating in our environment, and this is the major cause of the global health concern. Yeah, I agree, but I didn't study the any homologous same pattern. But if you check some other antibiotics like aminoglycoside, is the same conditions. It is the same way it is modifying and it is creating the global health concern. In other domain, other group of the antibiotics like aminoglycoside.
Okay, thank you. If anyone else has a question, please feel free to drop it. I have one. Yeah, I like it, like how you show us the huge diversity that was unexpected and was supposed not to be that diverse in other branches of the tree of life. However, in the part of the function, just because I'm unfamiliar with the research, I don't understand when you say that it's more with this trees.
Yeah.
Like the scales correspond about how effective is an enzyme with the same antibiotic that is the target, or also whether they are diverse in the targets in the antibiotics that they can break.
Could you please repeat your question?
Yeah. If you clarify a little bit more like what this log function scale means. It's more like there is an antibiotic and you test how effective the different enzymes are with the same antibiotics,
or
the diversity of antibiotics that they can break up.
Okay, I'm going to explain this tree again, and it will be more clear. And actually, in this scale, I set this scale from 0 to 1, and especially this blue color and this green or aqua blue is indicating the susceptibility, the moderate, resistant, respectively. And the red color is indicating the highly resistant. So if you check this tree, so here TEM 155, this is showing the almost resistant. And here some CT exome sequences are showing resistant. So here functionally both are same. Here sequence TEM and CT exome, both function are the same, resistant to Cefepime bacteria. Cefepime antibiotics, both are resistant, but their amino acid sequences are dissimilar. That's why they are located in different clade of the phylogenetic tree. So it means that if sequences are dissimilar, so it does not mean that their function will be different. So in this case, sequences are dissimilar, but their function is same. In bioinformatics field, generally we know that if not only the beta-lactamase enzyme, but in generally we are saying that if two sequences are same, then their structure and function will be same in bioinformatics field. But here we can see that two sequences are more than two sequences here, TEM 155 and 122, 121. This sequence is dissimilar to CTX in case of genotypic. But in phenotypic, this color coding is indicating the phenotypic data. And based on the phenotypic, these sequences are the same function. So, and these two functions are dissimilar from their ancient, here ancestor, this clade, major clade. That's why we are saying that these two enzymes have evolved through convergent manner. And here, in this is the case of resistant, but in case of susceptibility, we can check like here, the HN2. This is showing the susceptible, this sequence. And here, the remote located sequence like VEM. These two sequences are located in different clade. It means that their sequence is dissimilar, but their function is same, susceptible.
Okay.
So that's why this is a convergent manner.
Thank you very much. Much clearer. Thanks. And we have two questions.
And one more the general things I'm telling you, like a lot of bacteria, they are producing beta-lactamase enzyme. So bacterial species are different, like Klebsiella pneumonia, Streptococcus pneumonia, and E. coli. All these are different species. They are producing the beta-lactamase. But all these beta-lactamase are doing the same, like all these bacteria's beta-lactamase function is the same, like they are resistant to the different antibiotic, or they are susceptible to the antibiotic. Means their species is different, but they are functionally they are the same, are different. That's why in species level they are also doing the convergent manner evolution.
Thank you. So we have two more questions. We'll
Please.
one, like, did you check different mutations and variations in beta-lactamase in any clinical isolates?
Mutation related to mutation I didn't study, but as I presented one paper of Didier Raoult, and they selected the sequence from the archaea and the eukaryotes, and based on the wet lab experiment, they tested that the beta-lactamase is available or present in archaea and eukaryote. Based on wet lab experiment, they tested that it is present in the domain eukaryote and archaea. But related to the mutation and variation, I didn't check. But based on the in silico data analysis, I checked the amino acid residues, and according to the amino acid residues with the help of some Python program, I found that only one or two variation is not responsible for the complete functional differences. Means if two sequences are dissimilar, only one residues, then their function is not completely different. It is the same, like this.
Okay, thank you. And a last question says, you said that metallo-beta-lactamase were more diverse. Can you explain why? And if so, is this anything related with to histidine, the histidine they have in their active sites?
Yeah, actually, this is currently like we are studying the all family of the beta-lactamase class A, C, and D, and B. So we found that the beta-lactamase is a more diverse. Why we are saying that more diverse? Because presently I didn't study class A, C, and D completely. That's why we are saying that the beta-lactamase enzyme, metallo-beta-lactamase is more diverse compared to the serine beta-lactamase. Another thing is, yeah, of course, like because of the zinc ion, it is showing the similar function in different domain of the life also. And one more thing is like, even though the beta-lactamase is present in archaea and eukaryotes, but if you check their targeted cell, then compared to the bacteria, the structure of cell structure of the archaea is completely different. There is no peptidoglycan structure, and this is really a major problem in the scientific community, that if beta-lactamase is present in archaea, but there is no targeted cell, so how it is working in different domain. So this is the major concern in our scientific community, but still we are unable to answer the complete question, but still it is going on in our group. Yeah.
Vivek, thank you very much. It was a great talk, and what a way to open the symposium. If anyone has like additional questions, you still can drop them in the Q&A, and Vivek and the other speakers will monitor and they can reply back there. So before going to a short break of 5 minutes break, we are going to drop the information about how to become a member of the Society for the Study of Evolution through the Global Assistant Program. If you are interested, the information will be available in the chat. So thanks again, Vivek.
Okay. Thank you very much for scheduling this discussion. Okay.